So in this video we're going to continue our discussion of carbohydrates from a chemical perspective and at the level suitable for a GOB class, an introductory general organic and chemistry and biological chemistry class. Okay, so Benedict's reaction is a redox reaction, and this picture here actually is probably what you would see in the lab, where the blue would be a negative result, and these would be positive results. Okay, Benedict's test is a redox process, so let's take a little peek at how it works. So I'm going to pull these slides over. Definitely Benedict's is something I would like my students to know how to do. So let's just take a second to look at it. So Benedict's test for reducing sugars. For the most part, I would just say to my class that reducing sugars here means an exposed aldehyde. Okay, so aldehyde, we know what that means. What do I mean by exposed? By exposed, I mean Fisher projection. So I do not mean Haworth, so it has to be an open linear form of the monosaccharide. Okay, so let's have a look. So let's say we have our sugar, and let's say that the sugar, we'll just draw an aldehyde because that's really what we need. So let's say this is our monosaccharide. Or it could be anything, you know, it could be a different type of sugar, but the essential part we need is the aldehyde. So let's say it's a monosaccharide, and we add Benedict's. Benedict's is a basic solution of copper 2. Now copper 2 is blue, so we may as well use the blue pen. So it's an aqueous solution of copper 2 ions at a pH greater than 7, and it has a blue color. Okay, let's go back to actually just for argument's sake, let me make this black because I'm using blue and red, so let's go ahead and make this all black. So aldehyde, let's say it's a monosaccharide. It's our exposed aldehyde, it's a reducing sugar, it's in its Fisher form, etc., all that good stuff. We add blue Benedict's, and the active ingredient in Benedict's is this copper 2 ion. Well, what happens is we oxidize the aldehyde and reduce the copper. So what we get is we oxidize the aldehyde to a carboxylic acid. Where have we seen that chemistry before? We saw that chemistry in the previous video. Primary alcohols are oxidized to aldehydes. Aldehydes are oxidized to carboxylic acids. So this is a carboxylic acid. pH is now less than 7. And I'll switch back to the red pen. Copper 1+, which is a red-brown color. Okay, so this is a redox process. See how effectively I'm clicking on. There you go. So it's a redox process. And essentially what we've got is the monosaccharide is oxidized, and the copper is reduced. So the whole process is redox. So I would like, again, to emphasize to my students that I would like them to know this process. It could be an assessment question. And importantly, you get this characteristic color change from a blue color to a less blue color. And in reality, it could be anything in between. It just depends how extensive the redox has been allowed to occur. So to move away from blue, it could be green. So if you see essentially anything that's moved away towards the red end of the spectrum, so along the way we have, let's see, so from blue to red, don't be surprised if it stops along the way. So there's orange, yellow, and green in the middle. So if it looks more orange, it could look green, it could look yellow, it could look orange on its way towards looking reddy brown. So be on the lookout for that as well. Okay, let me engage this board again. Alright, so let's get back onto these slides. There you go. Quicker I move the slides, the shorter the video. Alright, let's try and get back over. There you go. Okay, disaccharides. What's a disaccharide? Well, di means two. So a disaccharide is a collection of two monosaccharides joined together. So a disaccharide is a monosaccharide joined chemically to another monosaccharide. Okay. The bond here is a glycosidic linkage. And there's different carbon atoms that can be involved in the glycosidic linkage. And if we get more and more monosaccharides joined together, so let's say this monosaccharide is a blob, and we can link that blob with another blob, then you could have a blob and a link and a blob and a link and a blob, where each blob is a monosaccharide. And eventually, if you have enough of them, let's say we have n of them, that would equal a polysaccharide. Where poly just means many. Many what? Many monosaccharides. Now a polysaccharide is also known as a complex sugar, and these are the good sugars, or at least so the reputation goes. Monosaccharides and disaccharides, so disaccharides and monosaccharides, these are also often called simple sugars. And these have a bad reputation. Do they deserve that reputation? Grr, that's my grr face. This one's got a patch over its eye. I'm going to make this a pirate. Look, it's got a patch. That's supposed to be a patch. It looks like a cap now, but it's supposed to be a patch. Anyway, so this idea of good and bad, is it scientific, or is it just hearsay in social media? Well, let's have a look. Let's play out our prejudice. Alright, that slide looks like we don't need it. So I'm skipping over some of these slides. You know, my students have these slides. They just tell you some important stuff about disaccharides. We really don't need it. Let's see if they have anything that we should probably pause over for a second. It looks like the next few slides are just looking at some vocab, so I'm going to push them off to one side and we'll clean slate. Okay, so let's spend a few minutes talking about these structures. So I guess the question I want to ask my students is, are the labels, comma, good versus bad, relevant to sugars? And the answer is, it's complicated. So I'm sure you know, reasonable people know that chemicals don't have the capacity to be good or bad. What makes a chemical good or bad is the behavior of the consumer of the chemical during consumption and before consumption and after consumption. So an example of why it's complicated: simple sugars, the bad ones, are found in fruit. And fruit is good, right? Yes, but eating too much fruit can lead to obesity, and that's bad. So fruit can be bad if you eat too much of it. So simple sugars are good if you eat some of them. They can be bad if you eat too much. Complex sugars, these are the good ones. Are found in starchy foods. So that's why they're good. So starchy foods are good, but eating too much of them can lead to obesity. Too much of them can lead to obesity, and obesity is bad for your health. In summary, it's the eating too much of anything can make it bad. Okay, so, in my opinion, I would definitely encourage my students to question these labels that come a priori with chemicals that have no business having labels attached to them. And for the remainder of this GOB course, I will try and link, as we start to look at biologically relevant molecules, I'll try and pick out these labels and how they are imposed quite unfoundly by people, and they should be questioned. The validity of these labels should be questioned. Okay. Okay, so let's get back to the slides. I do believe there is a diagram I can use coming up. So yeah, it's worthwhile looking at this slide, I think. So polysaccharides are these long chains of monosaccharides. These are the good sugars or the good carbohydrates. They can be incredibly long, tens of thousands of monosaccharides long, all connected via glycosidic linkages. We've all, see, I've lost my pen. We've all heard of these words, starch. Let's get rid of this pen. I'm going to go back to my red pen. If I can figure out how to activate my pen again. Go back. There you go. Let's get the red pen going. So starch, glycogen, and cellulose. Starch, you can definitely digest this. This is readily digestible. Glycogen is storage form. So essentially glycogen is storage for animals. Starch, you could think of it as storage for plants. We eat the plants, and it's a digestible form of starch for us, a form of polysaccharide for animals. And cellulose, this is the indigestible. Gestable. I'll probably regret spelling it with an A at some point when I realize it was an I, but oh well. Indigestible material that I guess has the property of adding to our fibrous intake. Okay, so let's see, based on what the next slide says, I might have to detour again and look at some structures. I'm hoping that they have some. I'm hoping that I can also move this over. Let's see. Starch is the most important source of carbohydrates in the human diet, and accounts for more than 50% of our carbohydrate intake. Yeah, it's very important to eat starch. You know, one of the problems people have if they go too crazy with a protein-rich diet, they can stress their organs out by not having enough starch. So it's always a good idea to have a balanced diet. That goes without saying. Right, let's look for a structure on these polysaccharides, because words are great, but structures are better in a structure class. Finally, here's a structure of, or at least a piece of a structure of a polysaccharide. So we're told on the box this is amylose. Okay, so let's inspect the structure. Well, first of all, notice that we have represented here, we've got the word etc., so we assume that this would carry on ad nauseum to a much longer structure. Okay. Notice that we have our nice monosaccharides, so these would be, well, we recognize that these are Haworth projections. Of our monosaccharides. Notice if we label, they've started to label some of these carbons. We've got carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So I'm just labeling one of them. We do the same for an adjacent one, carbon 1, 2, 3, 4, 5, 6. So you'll notice that carbon 4 is connected to carbon 1. So that's why they say it's a 1,4 linkage, and we know it's between two monosaccharides, so it's actually a 1,4 glycosidic linkage. Okay, it's a 1,4 glycosidic linkage. What else can we say about this? We know, if I look over here where it's a bit less cluttered, I know that there would be an H stuck up here. There would be an H down here. Typically we don't draw these H's, but we know that they're really there. There'd be an H up here. So don't forget those H's are present. Notice that, remember our acetal and our hemiacetal. So remember, an acetal had a carbon with two ORs on it, and we said because of that structure it was an acetal. And then if we had just one OR and one OH, that was half of an acetal, so we call that a hemiacetal. Okay, a hemiacetal. Well, that's exactly what we have here. We have acetals. So essentially, if I just draw a quick cartoon, I'll make this the last one. So let's say we go from left to right, the way it's drawn on the page. Then the rightmost monosaccharide here contains the hemiacetal. Because of its anomeric carbon here with its OC, OH. But all of these internal structures in short, the ones that are not immediately on the right end, these are all acetals. Now, what's important about that? Well, my goodness, what isn't important about that? This is partly where this idea of good and bad sugars comes from. So I'm going to try and drag this up. I've asked my computer several times to drag it up now. I guess you have to ask and then record yourself asking before it actually moves. Okay, here we are. So let's move to black just to have a bit of contrast here. So in terms of stability, acetals have a greater stability than hemiacetals. Which have a greater stability than aldehydes. So we've seen already that alcohols can attack aldehydes because of their exposed carbonyl. Remember, the carbonyl is that carbon double bond oxygen. So we can actually protect an aldehyde by making it a hemiacetal, and we can further protect the hemiacetal by making it a full acetal. So if we look at this sequence of polysaccharide here, this is less reactive, acetal, sorry. This is less reactive. But then this is more reactive. So the hemiacetal is more reactive in the sense that it's capable of opening up into its Fischer form. So these acetals are that essentially they're trapped as Haworth projections. But the hemiacetal is able to form the Fischer form. So the hemiacetal is able to open up. Essentially, you break this bond here, and then you've got your carbon 1 and your carbon 5 again. So again, the hemiacetal is free to open up, form its Fischer form, and what can happen with a Fischer form? It can be attacked by Benedict's reagent. So if we were going to test for the presence of a reducing sugar here, the only sugar in this example of the polysaccharide here that would react is the terminal carbon here. So this is essentially is the essence of the difference between, or the goodness in a polysaccharide versus the supposed badness in a monosaccharide. So let me see where I can maybe talk about this a little bit. Yeah, let's mention this real quick, because then the slides are going to wrap up by looking at glycogen. So I want to get ahead of glycogen. Okay. So let's try and do this as effortlessly as possible. That's a word I can't say. There's a lot of effort goes in saying that word. Okay. Right, here's where my cartoons get a bit more lazy. But again, it's just a cartoon. So let's say this is my polysaccharide, where, just to orient you, this is an oxygen atom, but this is the Haworth projection. So this is a cyclized sugar, and it's just connected via an oxygen. Okay, and it goes on ad nauseum over here. Okay, so it's our polysaccharide. Oh, keep going, you can do it. It's our polysaccharide, and polysaccharides are good, or at least that's what they would have you believe. Okay, let's terminate this with our hemiacetal. So again, only this bit is the hemi. And these are the cats, are all acetals. Okay. So if you've consumed this material and you are a hungry bunny, then your body wants to start metabolizing the sugar. And eventually in subsequent videos, we'll look a little bit at metabolism. We're not going to do it in this video, otherwise it will never end. But eventually you're going to want to start metabolizing this. So you essentially can only metabolize the hemiacetal. And there's only one of them. So although you've got a big, long structure, you've essentially only got access to one of them at a time in our little cartoon thought experiment here, at least anyway. Okay, so essentially, if I can get my little red pen to select red pen now. This is not a good commercial for... Oh, there's a little button, maybe I have to press that. There you go. Okay, so if I break this bond here, I'm left with 4 units, and I've got an extra unit here. So I can use this, so this now is used for fuel, gives me some energy, and I can do the same again to the next one. I can break this and use that for fuel, but notice I can't use them all at a time. I can only use one at a time. So I can use one monosaccharide at a time. So what does this mean? This means that the change in the concentration, square brackets, of monosaccharide is small. And that's good because we evolved for these small changes. We evolved to eat starchy sources of sugar. We also evolved to eat fruit, but the type of fruit that we evolved to eat were more like small berries than large watermelons. So our ability to find massive quantities of small berries probably wouldn't have exposed us to massive surpluses of simple sugars in our natural environment. So this is good. This is polysaccharides, access, no matter what you eat, access to one at a time. So I like to liken this to, in terms of a sugar dose, this is like a slowly leaking faucet. It's a dripping tap. It slowly drips monosaccharide into your system. Let's look at the bad guy. Let's look at the bad simple sugars. Well, as you might expect, here all the linkages are broken. So all or most, depending on whether you have mono or disaccharides, the glycosidic linkages are already broken, which means you have ready access to a large concentration of monosaccharide. Which means that when you consume this, the change in the concentration of monosaccharide is large. And it can often be greater than your body's tolerance for sugar. So why can that be bad? Well, let's say this is time, and let's say this is the concentration of sugar, and let's say your brain needs 5 millimolar to stop you from dying. Then you could, at this event here, you could eat a healthy meal. So let's say this contains polysaccharides. And there's a fluctuation in your blood sugar levels, but it's within tolerance. Maybe another healthy meal here. And you've got hormone-regulated sugar level, so your blood sugar goes up. Insulin helps it come down again. Maybe your blood sugar goes down too much, so then glucagon, another hormone, can help. So a mixture of insulin and glucagon can help regulate your blood sugar back to a normal fasting level. But then, boom, here, at this time here, you ate candy, lots of candy. So these are simple sugars, mono or disaccharides. This might be far beyond your tolerance, I guess, depends how much you eat. But this would be a sugar rush. And your body immediately attacks the sugar with insulin because you're about to die. Your brain's about to jump out your skull. But there's an overcompensation. Like in most alert situations, you overcompensate. And now you've got a sugar crash. Now you're about to die again, but for the other reverse reason. So now your body secretes glucagon, but too much. You're going to rush again, and so all of this is going to stress your body. So this can be bad. And this is one of the reasons why, school-age children should have a nice, healthy meal before trying to learn something. Can you imagine if, this could take several hours to play out. Can you imagine if this is like 8 a.m. on the way to school, and this is, 3 p.m. When the learning has just not happened for the 5th day in a row. So it's very important to get a nice nutritious meal, especially when you're trying to learn something. So again, anecdotes aside, link this phenomenon back to the change in concentration of monosaccharide. You don't really get this sugar rush from the complex polysaccharide because of all those acetals that are not readily available for metabolism. Whereas you do get them, if you scroll back at your leisure, you do get them when you see more of an abundance of these monosaccharides from a simple sugar. Okay, so I think we've said enough on that. To say more would be to repeat the same thing, and that's the whole point of a video, right? You can just pause it. Okay. I did want to have a look at just 2 more structures. So that essentially would be complex sugars. We'll assume a starch. They're the readily available ones to animals. Glycogen, this would be a branched form of a complex sugar, mainly for storage in the liver. So the only thing I want my students to be aware of is this highly branched form so it branches. When I say branch, I mean just coming off a, not linear. So this would be a linear segment here, but clearly there's a branching event here, and we get this like fork in the road type structure here. This is a highly branched form of a polysaccharide, is known as glycogen, and this is essentially how animals store energy for a rainy day. When a meal is missed or an animal, a hunt goes wrong, this is how animals can survive to hunt again. So I just wanted you to be familiar with that. Again, let's just inspect these labels here. Alpha, we know it's alpha because the oxygen here points down. Remember we said if the hemiacetal has an oxygen down, oh, I have no idea what happened then, but let's undo it. Nope. Oh, now I'm just making a mess. Okay, so let's try and get back. So the anomeric carbon here, if the OH is down, we said it's the alpha form, and the anomeric carbon here, if the OH is up, so an up OH we said was beta. So we look here, we've consistently got a down O, down, down, they're always down. So these are alpha glycosidic linkages. Okay, that's important because we're going to see beta in a second. As soon as I can engage this board. There you go. Nope, I had the feeling it was going to do that. So I just want to pull the whole thing down. I was hoping for a structure of cellulose. I think I'm going to get one, but I think I have to go even further down. No, there we go. I think the computer's just tired now. Okay, let's try and get this slide. This is one more slide I want to look at to kind of wrap up this video. And I think my computer's just on some kind of siesta right now. Okay, I want to go down. Let's see. Yeah, it's an oddly unresponsive screen right now. Let's see if we can salvage the screen. There we go. I was hoping for a structure of cellulose. I think I had a structure of cellulose when I last looked for it. Yeah, it's way down here. Okay. So that's all I want to do, computer. I just want to look at cellulose for my students before we wrap this video up. I'm pretty sure this is my computer, not the software I'm using. But as the saying goes, never work with animal children or technology, and you'll be fine. So become a farmer. Okay, that's why farmers are happy. Okay, so let's look at cellulose. We know that cellulose is another type of polysaccharide. Some animals have capitalized on metabolizing it, unfortunately. Think of termites. So in California, where we are bound with wood-eating termites, we decided to build living structures out of wood. Maybe in other states they build them out of sugar cubes. I don't know. But I never figure that one out. But anyway, look at cellulose. You and I can't digest cellulose. One of the reasons we can't digest cellulose is the beta glycosidic linkage. So notice here the oxygen is up. So we have beta anomers here. that are not readily digestible by you and I, but are readily digestible by termites and other insects that can digest wood. So this becomes a source of fiber for you and I, a source of fun and pleasure for a termite, and nutrition. So that essentially wraps up this video. We kept it under an hour. If I post another video on carbohydrates, it was because I felt my students needed something else. If I don't, this will be it for this carbohydrate tour for GOB chemistry class.